Okay, so we talked last week about the output zone of one neuron. The axon terminal. Action potential travels down to the axon terminal. Calcium rushes in. We have what are called vesicles. And those vesicles dock and they release neurotransmitter into the synapse. At the other side of the synapse is the postsynaptic membrane, which has channels, and those channels have specialized parts that are receptors that are designed to receive input from the neurotransmitters. And so the neurotransmitters bind, and the ions, not the neurotransmitter, but the ions travel into the cell. So in this case you might have potassium, sodium, any of these things. So neurotransmitters are doing the job of communicating from the output zone of one cell to the input zone of another cell. Okay, so there's ligands, which are sort of this lock and key type of mechanism that are designed to fit particular receptors. So remember, receptors on the postsynaptic membrane, they're receiving information. In the case of neurotransmitters, for example, these are considered endogenous ligands, which means that they're made from within inside the body. We have drugs and toxins. We talked about a couple toxins last week that bind to sodium channels, for example. These are what you consider exogenous because they come from outside of the body. So when people take cocaine, for example, it's an exogenous ligand, which is designed to bind to the receptors for dopamine, which is an endogenous ligand. So it's important for you to remember that when a ligand binds to its site, so here we see a ligand and a channel. This whole thing here is what you consider a channel or receptor. And so acetylcholine is ACH. This is a neurotransmitter that we release in our bodies. Very important at muscles, for example. Acetylcholine comes, binds to a particular site on the outside of the receptor, or the inside of the receptor, but basically on the outside of the neuron. Here's our lipid bilayer here. And it's important for you to remember that, look what's coming in here. Okay, so what's going from the outside to the inside of the cell is sodium, not acetylcholine. Neurotransmitters don't enter the postsynaptic cell. They just bind to the receptors on the postsynaptic membrane. And then whatever ion channel the receptor is in charge of, opens or closes depending on what it's supposed to do, and that moves ion flow. So as you can see, the sodium rushes in, and we know that this would cause an excitatory postsynaptic potential where the cell would become slightly depolarized from its minus 70 or minus 60 state. Probably not to threshold level, but enough to bring the cell closer. And then if there's another input somewhere nearby, those would amplify and have some summation. So we just saw an example of an ionotropic receptor, which acetylcholine bound to the receptor and the ion channel opened, allowing sodium to come in. This is one form of receptor. Another form of receptor is where you have something called metabotropic receptor, which works through G protein or some other internal cell signaling pathway, where in the case of ionotropic receptors, when you have a channel, the receptor is built on the channel just like we saw before with the acetylcholine, and when the neurotransmitter binds to the receptor, something else like sodium rushes in. That's considered ionotropic. In the case of metabotropic, you basically just have the lipid bilayer, you have the receptor on the outside, and basically no ions move in, but somehow that signals for some other factor like a G protein to go and talk to the cell nucleus and perform some kind of function there. So these ionotropic receptors are very common. There's many different types. We'll see examples of some. And so the important thing again is that here the neurotransmitter is binding to the receptor. Neurotransmitter does not enter the postsynaptic cell. The ions are what travels from the outside to the inside of the cell. And so this is what is considered ionotropic receptor, and it's a fast acting. So the neurotransmitter binds, immediately the channel opens, ions flow in. If the ions are positive, you get an EPSP, excitatory postsynaptic potential. If the ions are negative, it would cause the cell to hyperpolarize and cause the cell to have an IPSP. So here's a case of a metabotropic glutamate receptor, or metabotropic receptor in general. The neurotransmitter comes and binds to the outside of the receptor, and there's no channel here, okay? But there's an internal mechanism where these G proteins, these internal cellular messaging systems, might go to a nearby channel, which does not have its own receptor, and might cause it to open. And so, in a way, that these are more powerful because they can go to the cell nucleus, they can control what happens at the nucleus of the cell, and they can travel to multiple channels and cause those channels to open, and so it might have a larger impact on the cell. But they're much more slow acting, so they're going to take a few seconds or a few hundred milliseconds for the G proteins to engage and go to other cells, but they're also longer lasting. So once the G proteins are released inside the neuron, they might take a spread in multiple directions. They can even turn on and off the production of proteins in the cell, cause the cell to extend its dendrites, for example. And so these are slow-acting, longer mechanism type of channels. And so these are basically the two main types of receptors that you have. One allows ions to come in, the receptor sitting right on the channel, those are ionotropic, and metabotropic changes metabolism of the cell. That's why it's called metabotropic, and activates the G proteins, which can control activity all over the cell. So it's real important that neurotransmitter, once it's packaged in vesicles, and is released from the presynaptic cell, especially in the case of ionotropic receptors, it's going to bind to the receptor and activate it. This should be a very, very fast process. It's important that the neurotransmitter goes away from the synapse area very quickly. So there's three mechanisms that allow the neurotransmitter to be taken out of the synapse. The first is degradation, and so there are many enzymes which are found nearby. They float around in the synapse, and their job is to consume and degrade the neurotransmitter so that it can't bind to its receptor. If you watch any commercial for drugs, you may have heard the term MAOI, and so often for depression, a long time depression drug was an MAO inhibitor, or is MAOI. MAO is monoamine oxidase. We're going to learn about the monoamine neurotransmitters in a minute, but MAOs basically break down monoamines in the synapse. So if you take the drug, it inhibits the breakdown of the action of monoamine oxidase and allows more neurotransmitter to be present in the synapse. This is the case of norepinephrine and serotonin, and that elevates mood in some people. Another mechanism is what's called reuptake, which means that the neurotransmitter has its own channel, and once it's released into the synapse, there's channels on the presynaptic membrane that allow neurotransmitter to come back into the cell and be repackaged. The other mechanism is transport. So the transporters are the function of the reuptake. So the transporters are these sort of channels or pumps that sit on the cell membrane, presynaptic cell membrane, and allow the cell to be taken back up, the neurotransmitter to be recycled and repackaged at the presynaptic cell. So we've been talking a little bit about neurotransmitters, and next we're going to identify different classes of neurotransmitters. So it's important to understand what classifies as a neurotransmitter. So the first thing is that it has to be existing in presynaptic axon terminals. So it has to be released from neurons and go into release from the axon terminal, especially when an action potential reaches the axon terminal. It also has to be produced within that cell. So each neuron has its capability of making one neurotransmitter. So it's important that we remember that every neuron makes only one neurotransmitter. So that means you are a glutamate neuron, or a GABA neuron, or a serotonin neuron, acetylcholine neuron. You're not going to have a single cell that produces acetylcholine and serotonin. Each cell has its DNA program that determines what neurotransmitter it releases, and it only releases one neurotransmitter. Other important things about neurotransmitters is that they have to do something. When they're released, they have to produce changes in postsynaptic potential. So they somehow influence the postsynaptic cell, whether it be directly through ion channels in the case of ionotropic receptors, or through metabotropic receptors. But it has to be able to produce some kind of change in the postsynaptic cell, otherwise it's not a neurotransmitter. And if you block the substance, if you block it from being released from the presynaptic cell, then you don't get that difference in the postsynaptic cell. And so these are some rules that, have to be used to classify something as a neurotransmitter. So here are some neurotransmitters. This is from the book, and sort of allows you to understand how the neurotransmitters relate. So basically these are related based on their family, which is really their chemical structure. We're not going to get in too much into chemical structure, but it's good, really, there's ones that I want you to be able to recognize and understand which ones go in which class. So you have your quaternary amines, like acetylcholine, which we talked about. We're going to talk a little bit more about acetylcholine in a minute, but it's really important for muscle function. It's also, we have a lot of acetylcholine receptors in the brain that are involved in learning and memory, for example, energy levels, things like that. You have norepinephrine, epinephrine, which is adrenaline, which really, adrenaline doesn't work much in the central nervous system. It has sort of a cousin, which is norepinephrine, which, again, as you might imagine, is involved in energy. Dopamine, we're going to talk a lot about pleasure and reward. Drug addiction, real important in that. All of this is in another table, so you'll get all this information in a minute. Serotonin is 5-HT, also involved in mood. Melatonin is considered a neurotransmitter. It's involved in energy levels in sleeping and waking. The two big ones that are really involved all over the brain are GABA and glutamate. And so these guys are sort of the essential nuts and bolts of brain neurotransmitters. GABA is inhibitory and glutamate is excitatory. GABA tends to open chloride channels on postsynaptic cells. Glutamate tends to open sodium channels on postsynaptic cells. So they have opposing effects, even though they're both from the amino acid family, and actually one is they're very related to each other in terms of how they're made. Then you have your opioid peptides, your enkephalins, your endorphins, your dynorphins. These are obviously endogenous opioids, and if you give opiates, these are the receptors that the opiates will bind. Other neuropeptides are things like oxytocin, involved in social behavior, substance P involved in pain, CCK is involved in, it's a very localized inhibitory. Vasopressin, also involved in social behavior. Neuropeptide Y, involved in hunger. We're going to learn about that when we do hunger stuff. Hypothalamic releasing hormones, we'll talk more about hormones next week. And then there are even gases that have potential to move across cell membranes and to engage the postsynaptic membrane. And so what I want you to be able to do is sort of recognize a few of these key ones and kind of understand which families they belong into. So the acetylcholine, norepinephrine, dopamine, and serotonin all belong into the amine family. It's just kind of they're related to each other, which shows you, remember we talked about the monoamines. Monoamine oxidase will eat all of these different neurotransmitters. They'll eat serotonin, they'll eat norepinephrine, they'll eat dopamine, and so a monoamine inhibitor is going to affect all of these different neurotransmitters. GABA and glutamate are highly related in terms of the way that they function, the way that they are made, but have completely opposite functions, where GABA is inhibitory and glutamate's excitatory. And the opioids belong in sort of a class called the neuropeptides, along with other neuropeptides. So just recognize basically acetylcholine, norepinephrine, dopamine, serotonin, GABA, glutamate, opioids in general, and the rest are sort of things that we'll address later on in the course of the semester. So acetylcholine, ACH, is an amine neurotransmitter. And when you think of acetylcholine, I want you to think of muscle activity. Acetylcholine is released from the presynaptic cell. Our muscles, every muscle in your body has a postsynaptic membrane on it that is dense with acetylcholine receptors. When acetylcholine is released from the neurons that communicate to muscles, so think about flexing your finger muscles, that information comes down from your motor cortex at the top of the brain, makes a single synapse in your spinal cord, and then your lower motor neuron talks to the muscle groups and make your fingers move, for air quotes, for example. So the acetylcholine is going to be released from those spinal motor neurons onto the muscle groups that need to coordinate for you to be able to move your fingers. And so acetylcholine is released, and in that case, most of those receptors are what we call muscarinic receptors. There's also nicotinic receptors, so maybe this name sounds familiar to you. So nicotine, coincidentally, binds to nicotinic receptors. Nicotine obviously found in cigarettes, tobacco. And so it binds to those receptors, and it causes excitatory effects, so postsynaptic excitatory effects. It turns out that the high density of nicotinic receptors in areas that are rich in dopamine cause an enhanced release of dopamine. So not to confuse the issue too much, but it turns out that at the presynaptic membrane, we also have receptors. So when an action potential comes down and calcium rushes in, for example, and neurotransmitters are released, you can actually have nicotinic receptors that sit at the presynaptic membrane, and if acetylcholine comes and binds to those, they're actually going to increase the calcium. Okay, so more calcium rushes in these cases. Sorry, a red arrow here. More calcium comes in and releases neurotransmitter, and so it can enhance the release of neurotransmitter. Many nicotinic receptors are found on dopamine neurons. This is part of the reason why nicotine is so addictive, because dopamine is a highly addictive neurotransmitter. Muscarinic receptors, again, are mostly, in many cases, metabotropic, and they can be excitatory and inhibitory depending on what they do. So serotonin is, if you think of muscle activity for acetylcholine, I want you to think of mood when you think of serotonin. So serotonin is, as it says here, implicated in sleep, mood, sexual behavior, and anxiety. So one of the sort of big drugs that came out in the 90s was Prozac. And so we'll talk about how Prozac works. So again, every neuron secretes only one neurotransmitter. So if you have a neuron that has serotonin, and that serotonin gets released and binds postsynaptically, in a lot of cases, in almost all cases, I can't think of a serotonin receptor that's not metabotropic. So I think serotonin is almost always metabotropic and not ionotropic, meaning that it's not going to open channels, but it's going to have G proteins which do the work postsynaptically. But anyway, Prozac, which was obviously a big drug, and there's many baby versions of Prozac, newer versions of Prozac. Prozac is what you call a selective serotonin reuptake inhibitor. Selective serotonin reuptake inhibitor, or an SSRI. There's a few different classes of SSRIs. There's something called tricyclics now, which affect serotonin transporters, but also monoamines, other monoamines like norepinephrine primarily, but also dopamine to a smaller extent. And so what these do are, we talked about transporters. One of those mechanisms where we get neurotransmitter out of the synapse is reuptake. Reuptake is accomplished by transporters that sit at the presynaptic membrane and allow the neurotransmitter back in. SSRIs, the I stands for inhibit reuptake, so they block the reuptake, they shut down these channels, and the result is that the serotonin stays in the synapse for longer. You still have MAOs, the monoamine oxidases, which consume these, but the reuptake is faster than MAOs, so you get an accumulation of serotonin at the synapse, and this helps people with depression. Serotonin, more serotonin at the synapse elevates mood. We're going to see serotonin is also involved in some hallucinatory drugs. So one of the initial problems with Prozac was that people were seeing shadows that weren't there. There was some mild visual hallucinations, and this was probably because serotonin can affect some of these sensory systems and give you hallucinations. So glutamate and GABA, as we mentioned, are real important. They're the machinery of the brain. So remember that glutamate is excitatory, always excitatory. GABA is always inhibitory. Aspartate and glycine are not as common in the brain. They're more available during development, for example, and in other areas, peripheral nervous system. They're there, but the glutamate and GABA are the predominant neurotransmitters in the brain. When we talk about all of those motor neurons that are sitting at the top of your brain, communicate to the spinal cord, those are all glutamatergic. We talked about the cortex, for example, that has 6 layers. We have pyramidal cells which sit all throughout the cortex, dendrites and axons that go to other parts of the cortex. These principal cells, or pyramidal cells, are all excitatory glutamate neurons. And so all the communication that's taking place from one part of the cortex to the other, anytime you have axons moving from one area of the brain to the other, especially from the cortex to the cortex, it's going to be a glutamatergic neuron. But there's also important cells that sort of regulate this activity. So these GABAergic cells, which don't look pyramidal, but they connect onto the nearby glutamatergic cells and help quiet them down. And so glutamate and GABA play an important role interacting with each other. So again, we have opiates, and these would be considered the endogenous forms of the opiates that people consume, like heroin, morphine, oxycodone, all of these opiate types of things. Those would be the exogenous versions of what we naturally make in our bodies. And so opiates belong to a class of neurotransmitter known as peptides. And so these, opiate peptides mimic opiate drugs such as morphine. But I don't like that term there, mimic, because it suggests that the morphine is the natural. But really the opiates, it's the other way around. Drugs such as morphine mimic opiates, the opioid peptides. So there's quite a few of these in the gut, for example, on spinal cord, in the brain. We're going to talk about a particular area of the brain in a minute called the VTA, ventral tegmental area. This is part of our pleasure center of the brain. So you might hear about natural endogenous opiate release. For example, you may have heard of runner's high. This is an example where somebody who's exerting themselves, and opiate systems are highly involved in pain systems. So somebody who's running a marathon or something, you get a release of endorphins, natural endorphins, which give you a euphoric experience, take some of the struggle and the pain away from that strenuous activity. So obviously running is not the only thing that can produce this sort of natural pleasure. There's many other ways, and also under high pain situations, endorphins get released to counteract that. So, as you know, morphine and heroin are big analgesics. They're big for treating pain. That's why they're prescribed. Your Vicodin, your oxycodone, all of these things are prescribed for treating pain because they're really effective at it. They counteract that painful stimulus. They suppress pain receptors in the brain all through the way that they work. So here's another table listing. Really, these are the ones that I want you to sort of focus on here. And these aren't in different classes, but they have associated effects with each of them. So, and then they have psychotropic drugs. So psychotropic meaning influencing thought or influencing behavior. And so these are examples of the drugs that affect each of these groups of neurotransmitters. We're going to talk. You guys are going to do a mouse party activity in a couple slides, and hopefully that will give you an ability to see how these drugs work. So when we talk about the endogenous ligand, endogenous ligands are almost always agonists. So they sort of perform the function, the normal effects of the receptor, okay? So the receptor is sitting on the postsynaptic membrane, the endogenous ligand binds to it, and then whatever is supposed to happen happens. If it's an ionotropic receptor, usually it means a channel is opening and ions rush in. If it's a metabotropic receptor, it usually means triggering G protein coupled receptors and causing G protein to cause function changes within inside the cell. When you have exogenous ligands, like drugs, they don't always imitate the activity of the normal neurotransmitter. Okay, so think about, for example, SSRIs that we talked about. Those would be what you consider an antagonist because they block the receptor, they block the transporter at the presynaptic membrane from working. So if it works in the same way as the endogenous ligand, we consider it an agonist. If it counteracts the effects of a non-endogenous ligand, we consider it an antagonist. So sometimes drugs can block the receptor. Sometimes drugs are called competitive antagonists, where if you have a receptor, think about we talked about the receptor being sort of a lock and key type of mechanism, right? So think about if you have a key that sort of fits a lock. In the case of an endogenous ligand, we have a particular ability to bind that receptor. But if we have an exogenous ligand and it's an antagonist, maybe it blocks the receptor and doesn't allow the channel open at all. And it doesn't allow the normal neurotransmitter to bind to the receptor either. So it sort of takes that receptor out of action, sometimes forever, but usually for a certain period of time. So the receptor binds the drug, the exogenous ligand that's made from outside of the body, and it does something opposing what it normally would do if the neurotransmitter were bound. So we'll see examples of this in the coming slides. So when you have exogenous ligands, let's say you have a postsynaptic membrane, and you have a certain amount of neurotransmitter, which we consider physiological, meaning that it's the normal amount that the body's used to. There's sort of this give and take about what happens to receptors on the postsynaptic membrane. So you've got receptors there, a certain number of receptors on the membrane, and in the presence of neurotransmitter, the neurotransmitter binds. In some cases, you might have a reduction in the amount of neurotransmitter. Think about depression, for example. One of the problems with depression is that there may be a reduction in serotonin release from certain cells. So the postsynaptic cell sort of likes a certain low level, a certain amount of constant activity. And so what happens is, I'm going to change my color here. When you have a reduction in serotonin released from the presynaptic neuron, the postsynaptic cell starts putting out more receptors. So they upregulate. Same thing happens in the case of an antagonist. So if some of these cells, the receptors sort of get blocked by an antagonist binding them and preventing them from working, the cell's going to upregulate the receptors. Alternatively, if you have a postsynaptic cell and it has a certain number of receptors, but if you take something like dopamine, which essentially floods the brain cocaine, for example, floods the brain with a synthetic dopamine. That's exactly what cocaine is, right? So it's sort of a synthetic type of dopamine. Also works on norepinephrine pathways. But it provides an excess of dopamine at the synapse. Ecstasy is a great example of this too. Provides a real excess of serotonin at the synapse. So you've got way too much serotonin, way too much dopamine. And so what the cell is going to do to respond is going to pull in some of its receptors. The cell is getting overstimulated. And so this is an example of what we call downregulation. So neurons downregulate if there's too much activity, too much neurotransmitter released, or if we're getting too much of an agonist drug. We pull in the receptors because we're getting overstimulated. In the case of too little activity, we upregulate and increase the receptors. This also happens in the case of an antagonist. So this slide shows a lot of the things that can happen sort of at the presynaptic cell, all the different things that drugs can do. So you've got these exogenous ligands, these drugs, that are capable of doing things at the presynaptic cell. So there are certain things that we already learned about that can affect the conduction of neurotransmitters. So, for example, you might have tetrodotoxin or brachitoxin, which block action potentials from traveling down to the axon terminal and performing the action potential. And that happens. So if you have tetrodotoxin, it's going to prevent the action potential from traveling, and then the neurotransmitter doesn't get released. There's also drugs that affect the synthesis of neurotransmitters. Some cells, there's a few drugs that you're going to learn about in a few minutes that affect the storage of transmitters in vesicles. And some of the real gangsta drugs go in and break up the vesicles so that there's sort of all of this neurotransmitter sort of sitting at the postsynaptic cell, not packaged in vesicles, which is, think about putting something in a plastic bag. It can't really harm or perform its function if it's sealed. And so vesicles sort of perform a long-term storage of neurotransmitters at the axon terminal. And some cells go in and break up all this neurotransmitter so that it's constantly being released into the synapse. So obviously there's things that we talked about that affect the release of synaptic transmitters. So nicotine, for example, we talked about, causes an influx of calcium and causes an increase in the release of neurotransmitter. That's one of the ways that nicotine works. We also talked about presynaptic receptors. And so that would be, that's what we just mentioned. The case of nicotine affects release. Inactivation of uptake, SSRIs. In the case of serotonin, Prozac prevents the uptake of serotonin so that more of it is located at the synapse. And blockade of transmitter degradation. We talked about MAOIs, MAO inhibitors, another form of antidepressant drugs, also designed to keep more serotonin at the synapse by blocking monoamine oxidase activity, which normally degrades the serotonin that's sitting at the synapse. So here are a bunch of actions that can happen at the postsynaptic cell. So again, you have the degradation of neurotransmitters, so I guess that's at the synapse itself. We talked about up and down regulation. So if you have an enhancement, an excess of neurotransmitter, or an agonist, exogenous ligand that acts just like the neurotransmitter, in the case of cocaine, you might have a reduction in the number of postsynaptic receptors. They get taken into the inside of the cell. If you have an antagonist, which blocks the activity of that, or if you have a reduction in the release, you then put out more receptors at the synapse. You can actually block receptors from working. You can activate receptors, so here it's talking about LSD, case of nicotine and postsynaptic cell, can do all these things. Or you can affect the second messenger system. So lithium has been shown to do this. Lithium is still a common drug used for bipolar disorder. And so it engages second messenger systems. It increases expression of something called brain-derived neurotrophic factor, which is a protein that helps neurons to grow, and seems like that's really effective in treatment of bipolar disorder. So there's all these postsynaptic things that can work, and these are examples of those. So this is an example of a GABA receptor, and just shows you the ways that different things can affect the same receptor. Okay, so you first of all have GABA. There's a site for GABA, the endogenous ligand, the thing that this receptor is supposed to bind to. And what happens is when GABA binds, it opens the chloride channels and chloride rushes in, again causing the cell to hyperpolarize, causing an IPSP, making the cell less likely to fire an action potential. But look at these other sites. So you've got your benzodiazepines, like Valium. You've got barbiturates, so a number of the barbiturate drugs. You've got picrotoxin, which is a well-known GABA agonist. There's other neurosteroids, which we can talk about at a later time, that binds to neurotransmitters. And look what binds here. So alcohol is capable of affecting these chloride channels directly. So it goes, travels into the chloride channel and can cause the pore to open. So the mechanism of alcohol, again, as you're going to learn in a few minutes at the Mouse Party website, is that it causes chloride channels to remain open. So this causes cells to be hyperpolarized, causes it to inhibit. Think about, so obviously in case of heavy alcohol consumption, things get slowed down, reaction time gets slowed down. This is why driving is so dangerous under the influence of alcohol. Your ability to respond to things as quickly is severely suppressed. Also, speech, slurred speech. Things are also inhibited that, your inhibitions are inhibited, right? So you got people dancing on the bar, for example, because the prefrontal cortex, the area that normally inhibits a lot of instinctual behavior, things that we might not normally do, those neurons also get highly inhibited with the consumption of alcohol. So in the case of addiction, it's pretty well accepted that it's through the dopamine. So when you think of drug addiction, obviously some drugs are much more addictive than others, but dopamine seems to be the sort of common element in all of these things. So there's what we call the reward pathway, which is the ventral tegmental area. So the ventral tegmental neurons project, we'll see this in the next slide, project to the nucleus accumbens, which is an area of the brain that's involved in perception of pleasure. And so what happens is, in many cases, these are dopaminergic cells. So you have these dopamine cells that are coming in to the nucleus accumbens from the ventral tegmental area. They're releasing dopamine. in the case of drug addiction, we'll see an example for heroin, or we can take cocaine because it's more directly affecting the dopamine receptor than heroin. Cocaine comes in and completely floods the synapse. We have what we call supraphysiological levels of dopamine, or at least the dopamine agonists that are inside the cell. The postsynaptic cell pulls in its receptors as a result. We have downregulation of receptors. And so when you go back the next day after a binge of cocaine, you have the case where the dopaminergic cell now releases its normal level of dopamine. But because you have fewer receptors, many of the receptors were downregulated. And so the things that normally bring you pleasure in your pleasure center, things that make you happy on a day-to-day basis, interacting with friends, exercise, all these other day-to-day things that release a mild amount of dopamine, don't feel as good anymore, because a lot of these receptors that are there to sense them are brought inside, because they're so used to this other situation of having this excess of dopamine that now all their receptors are internalized. There's not many receptors on the postsynaptic membrane. So the body craves this level of activity that they had in the case of the drug. And so people go and find the drug, consume it again, and they go through this vicious cycle where day-to-day activity is not as stimulating anymore because the body's so used to this high level of dopamine activation. So here's a look at the human brain with the VTA, the ventral tegmental area, which is situated sort of right near the hypothalamus, top of the brainstem. Projects really strong projection to the nucleus accumbens. Also has projections throughout the frontal lobe, so a lot of our frontal lobe area is controlled by dopamine. Dopamine inputs control emotional things like emotional memories, particularly in the medial prefrontal cortex, so right in the middle of the brain here. There's a lot of dopamine inputs that control how we respond to situations, how different things make us feel. So obviously more than one thing people can become addicted to. There's examples of obviously drugs. Drug addiction probably the most severe form of addiction, but there is such a thing as sex addiction. Shopping, people become addicted to shopping. Exercise, video game addiction, big thing now. Gambling addiction. All of these different things have been identified to be related to this pathway of the VTA projecting to the nucleus accumbens, but also these projections. And just like I mentioned in the last slide, these things cause such a surge of dopamine that the other activities, the day-to-day activities that don't release this thrill-seeking type of activity level, don't do as much for us and make us want to crave that activity again. So this is an example of a rat brain, and so you can do this technique called microdialysis, where you can go to the nucleus accumbens, for example, put your probe in, and basically measure the level of dopamine. So you basically inject a fluid, it goes through, comes out the tube, and then you sort of analyze how much dopamine is in your thing. And as we'll see in the next slide, we know that basically drugs can cause a real surge in the dopamine production of neurons, and this is how we know people have mapped the addiction pathway, because animals become addicted to drugs just like humans do. So here's if we allow the rat that we just were doing microdialysis, if we give it cocaine, either into the blood, or in some cases you can give it directly into the nucleus accumbens, for example. You look at dopamine levels in the nucleus accumbens, allow rats to press a lever and give themselves an injection of cocaine. You see that in the case of saline, they really don't press the lever much to begin with. They press it just out of curiosity and might press it 2 or 3 times. But in the case of cocaine, if they are allowed to press the lever to receive an injection, they'll press it, and you can see the surge of dopamine in the nucleus accumbens. Again, this is how people have identified the pathway of a drug addiction. So these animals we'll start to press the lever as often as possible to get that rush of dopamine in the nucleus accumbens. So here's an example of the cortex. I think this is in an animal, in a rat that was received either a single or in this case a few injections of ecstasy. And so what we're looking at here, all of these white fibers here are all serotonergic axons. So serotonin normally has a high level of projection to the cortex. And so what happens is, as we talked about with the downregulation, the other side of the story is that if there's no receptors, there's also downregulation that occurs at the level of the axon. So the axon, if it finds that it's releasing, if it's not using, so ecstasy, as you'll learn, is working postsynaptically. It mimics serotonin, causes euphoria, and that's where its name comes from. And so what happens is serotonin, which is normally projecting into this area, the brain works much in terms of what we call negative feedback, where the cells here in the cortex communicate back to the serotonin neurons. Most of them are located in an area of the brain called the raphe nucleus. And so these cells are normally talking to those serotonin neurons saying, hey, give us more serotonin, we need more serotonin, please send more serotonin. When ecstasy is present, that feedback goes away. So those serotonin neurons that are sitting in the raphe nucleus are not getting any signals to release serotonin, and as a result, those cells sort of start retracting their axons back further and further and further because they're really not needed anymore in this area. And so again, the day-to-day in and out activities aren't as pleasurable for people who start to abuse drugs like this because they're not getting that normal release that they would from day-to-day activity. And this is, again, ecstasy is not considered a highly addictive drug because it's not working through the dopamine pathways, it's working through serotonin, which isn't as strong an addiction pleasure reward pathway. But still, the surge of euphoria that you feel under ecstasy is not going to be repeated, and now the day-to-day activities are not as satisfying anymore either. And so this is how long-term effects of ecstasy can be problematic in the brain.